So thank you. This work is in collaboration with the Crack Center where I belong and the IRTA Center that both are in the area of Barcelona. So we are interested in the study of variation within a species and also in the interaction among species. A way to study the variability is by using population genetics that is looking for understanding the levels of patterns of diversity, but also what are the mechanisms that are involved in this variability. These frequencies or the variability are affected by external factors, such as abiotic factors, for example, climatic change or disasters, but also by biotic. For example, well, with the interaction with other species, such as symbiosis or others. In the relation to when we want to look at the interactions of host and microbiota, so there were recent experiments that suggest that a reduction in host genetic variability constrains microbiota diversity. Here, what we can see in this first plot is that the host diversity using different populations with lower or higher diversity, so they detected higher microbiota diversity. The researchers hypothesizes that the host genetic variation can influence the microbiota diversity and both can influence the host fitness. So it is important to compare the levels and patterns of host variability with microbial diversity, but at an individual level, using every individual. When we want to study the variability of host individuals, so the classical way is that as you take a sample of the population, from here, this population, you take a sample, here you have individuals with their microbiota that is included with him. And then the only way to do that is by means of the heterozygosity. If in a diploid species, so you have two chromosomes, and then you can compare the differences, the average differences within an individual. But in fact, in a population, so we have more information related to the frequencies that are included. So, using classical population genetics, if you want to estimate the variability in a population, so you do genealogy, so you sequence. So this is the genome. Imagine that is a genome, and the red dots indicate a mutation that is in this sample. So the first one, this one is a mutation that is a frequency 1 in the sample. Here, there is a mutation that is a frequency 4 in the sample. So, with all this information, so you can obtain what is called the site frequency spectrum, that is a summary of all the mutations. So here we have, in that case, 18 mutations at frequency 1, that are all these, and for example, 4 mutations at frequency 4. And then we have a plot with the spectrum of the diversity. With this data, so we can estimate the variability. So using different weights, so we can focus on different aspects of the site frequency spectrum and then take different estimators of variability. And then with these estimators, we can allow to infer evolutionary process using tests like Tajima's-DA that compare the different estimators of variability. In our approach, what we did is just, here I show 2 examples, one for haploid and one for diploid. We focus on a diploid. So what we do is that we take the site frequency spectrum, but per individual, or per individual or per haplotype. So how we do that? So here what we do is that we take the frequency of each individual, but in relation to the whole population. This one is the site frequency spectrum for the whole population, and these are the site frequency spectrum for each of the individuals. So here we have a mutation that is a frequency 2 in the sample, so we include that number 2. Here we have no mutation, so nothing. And here we have a mutation at frequency 6 in the whole population, so we include 6, here 4, etc. So using this information, so what we can do is the same than before, but we have to wait by the probability that the mutations are not appearing always in one individual. So using this estimator, so we can also do the same. So we can estimate different estimators. We can take different estimators of variability, and then this data allow to infer evolutionary process, but using this relative side frequency spectrum. So as an application for classical population genetics, is just to detect, for example, spatial gradients patterns. Imagine that you are studying in an area, a plant, for example, Arabidopsis thaliana, that is a diploid selfing species, but is effectively an haploid species, and you take one single haploid genome for each region. So in that case, if we can look if this pattern, for example, is under panmixia or under if any other evolutionary process occur. So if we can do by simulations. In that case, if there is panmixia, so all these samples will show no pattern across all the area. So all these different estimators will have patterns that are uniform. But if there is a process of expansion or colonization, so what we can see is that taking just a single haploid sample per area, so we can detect patterns that using different estimators, so can allow to interpret what evolutionary process happened. The application to host microbiota is to compare the patterns of host diversity per individual and using their microbiota. So you have a sample of the individual and their microbiota. And then you estimate the variability and of the host and the diversity of the microbiota. So we can try to see if there is a pattern between these diversities. But at genome level, the evolutionary events have to be very strong to see these kind of patterns. But these estimators can also be used to detect association of host variability with the microbiota diversity at the specific regions of the genome. That is what we call signatures of holobiont diversity. As an empirical application, we use Duroc pigs from a farm. And then what we saw is that at genome level, if we compare the host diversity and microbiota diversity using different estimators, we don't see any pattern. That is what, in fact, it is expected because we are not expecting strong differences in a farm population genome scale. Nevertheless, we can see that if we study the association at the specific genome regions, we saw using this what we call diversity-wide association study. So we detect several genes that are associated to mucosal immunity and other functions that are related to the host microbiota. So in summary, so we have developed a method using the relative side frequency spectrum to estimate the host variability. This method can be applied to population genetics field, but also to the interaction of host microbiota. And an empirical analysis found candidate genes that can be implicated in the interaction host microbiota. Okay.
Thank you. We have time for a quick question, if anybody can beat me. Okay, I'm going to ask the question. This is actually fascinating. I once wrote a grant on this and got told I was very wrong for studying this correlation. And what kind of data do you need whole genome sequencing for this, or can it be applied to SNP chip data? Or?
Yeah, well, for estimating the variability, we need the genome information because you cannot use only genotyping because the variability is like a bias.
Super. Yeah. I look forward to talking to you more later. No one else will get the chance, I'm afraid, so thank you. I'll talk to you in the break.